I'm Daniel Levine, and this is the Bio Report.
Checkpoint inhibitors aren't new, but Immutep is developing therapies that target LAG3, a checkpoint discovered by its chief scientific officer. The LAG3 gene codes for a protein that plays a role in the regulation of the immune system and is expressed on T cells. Immutep is seeking to target LAG3 to both stimulate the immune system to treat cancers and suppress the immune system to treat autoimmune diseases. We spoke to Marc Voigt, CEO of Immutep, about the LAG3 checkpoint, the company's lead experimental candidate, and its partnerships with GSK and Novartis to develop therapies for cancer and autoimmune diseases. Marc, thanks for joining us.
Pleasure. Thank you for having me.
We're going to talk about Immutep, its lead experimental therapy, and the target it's pursuing as it seeks to address cancer and autoimmune conditions. The complexity of the immune system seems endless as new targets to regulate the immune system continue to emerge. How well are we able to modulate the immune system with existing therapies today?
Yeah, first of all, there are some pretty good therapies out there. Cancer immunotherapy or immuno-oncology has been changing the treatment landscape. First of all, it really started with anti-CTLA-4 back in 2011. Then very, very successful development of the anti-PD-1, 13, 14, and especially the anti-PD-1 therapies have been super successful. In the past 2 decades ago, maybe it was seen a little bit, as a joke or a kind of fantasy that your own immune system could help to eradicate cancer. And then you saw with cancer immunotherapy that actually a kilo of lung tumor could simply disappear based on your own little T cells. So there has been a great deal of success. And in many different indications, 30 indications or so, you saw important changes. However, on the other hand side, since 2013-14, there have also been a lot of disappointments or misfortune or targets where the initial hope didn't materialize finally. So it took 7-8 years actually to have the next one, which is LAG3, clinically validated last year and commercially validated and approved this year by the FDA in March and also approved in Europe. So a great deal of success, but also a number of concepts which didn't work out.
Immutep is focused on LAG3, which is a cell surface molecule. What role does LAG3 play in regulating the immune system?
Yeah, LAG3 is an immune checkpoint, and you can think of these immune checkpoints like little triggers in our immune system with which you can dim the immune system up or down. Obviously, in oncology, if you have cancer, you would like to have an active immune system, actively fighting cancer. In autoimmune system, your own immune system is obviously, in autoimmune diseases, your own immune system is obviously out of control, and you would like to bring it back to the right balance. And LAG3 is ideal, we believe, for both approaches. On the one hand side, you can target the T cell, if you like, the soldiers of our immune system, CD8 T cells. On the other hand side, via the major ligand MHC class 2 on the antigen presenting cells, dendritic cells, monocytes, if you like, kind of the generals of the immune system, providing the information whom to attack and also heavily involved in drafting new soldiers, if you like that analogy. With that, you can activate the immune system. So there is a negative modulation and there is a positive modulation of the immune system. So the LAG-3 is one of the great targets in immuno-oncology as well as in autoimmune diseases.
And how common is LAG-3 to these cells? Is it how specific a response can you get with it?
You can get specific responses in autoimmune diseases. These T cells, and LAG-3 is an exhaustion marker, accumulate at the disease site. So it's very specific there. It's a very upstream approach because the T cells are the basis practically for more than 90% of autoimmune diseases. So that could be pretty specific. And in oncology, there are different approaches. On the one hand side, anti-LAG-3, where you keep your T cells active, similar approach like anti-PD-1, anti-CTLA-4, anti-TIGIT, you name it. So you block the checkpoint in the T cell in order to keep the T cell active. And on the other hand side, with activating the antigen presenting cells via MHC class 2, you have an option to broadly, if you like, boost your immune system, so boost your antigen presenting cells, activate them, and then via the physiological way have more CD4, CD8 T cells, more NK cells, and also a Th1 response subsequently.
There's this growing arsenal or tool chest to stimulate the immune system, calm it down. What makes one approach better than another for a given disease?
The data, at the end of the day, it's evidence-based medicine. So you need to have the data generated at the end of the day in clinical trials, as nice as preclinical experiments are, and they give some hints, of course. At the end of the day, it's about clinical research, and you need to be either superior in terms of efficacy, or you need to be as efficacious but better tolerable. So you simply have to play a role in the concert of standard of care. As simple as that. It sounds simple maybe, but it's of course a big challenge. You have a variety of different clinical trials, combination with anti-PD-1. There are hundreds of clinical trials out there. So you have to differentiate yourself, and you have to convince as a concept, irrespective what kind of concept it is, with clinical data.
Your lead experimental therapy is known as FT. What is FT and how does it work?
FT is practically LAG-3 itself, if you like. So we use the checkpoint. We use LAG-3 to activate the immune system via MHC class 2. So it's an antigen presenting cell activator, an APC activator. So it's distinctly different from the anti-LAG-3s. And when LAG-3 is being discussed, very often one is automatically referring to anti-LAG-3, which is done by Bristol Myers Squibb, by Merck, and by our licensee Novartis. But eftilagimod is a unique APC activator. Actually, we are globally the only ones activating the antigen-presenting cell via MHC class 2. There is TLR, there is CD40, there is STING, and maybe the one or the other concept. But I believe we have here the most powerful and least inflammatory way to activate the APCs. And our leadership position can maybe also be explained by the work of our Chief Scientific and Medical Officer, Frédéric Tribel, who actually discovered LAG-3. He is an immunologist and oncologist and the worldwide leading scientist for LAG-3. So we have all our programs centered around LAG-3, and the center of gravity of what we are doing is, as you rightly said, eftilagimod or FT.
You're exploring this in combination with other immunotherapies, with chemotherapy, and with both. Why are you not looking at FT in cancer as a potential monotherapy?
Oh, we did that. So we have some monotherapy experience, for instance, in renal cell cancer. There has also been different scheduling or administration work being done as a monotherapy. We have, if you like, kind of IO monotherapy trial in metastatic breast cancer, where we combine eftilagimod with chemotherapy versus chemotherapy plus placebo, but FT is obviously the only IO. So there is some monotherapy data and, of course, activity. But on the other hand side, the reality is that in most indications, you actually face combinations. Let's look at, for instance, non-small cell lung cancer. You even have their doublet chemotherapy as one standard of care, or Keytruda plus chemotherapy. And combination therapies are established since decades. FOLFOX, FOLFIRI, for instance, in colorectal cancer. So it's nothing new, and I believe it's also the right way to go, to combine with different standard of care. So to provide the patients with hopefully good tolerability, with the best potential outcome. And this is typically achieved in most indications via different combinations.
What makes it a compelling choice as a combination therapy?
First of all, it's an easy route of administration. So eftilagimod is given subcutaneously, 30 milligram, unlike, for instance, immune checkpoint inhibitors, anti-PD-1, often given intravenously, 200-300 milligram or up to 800 milligram, depends, of course, on the concept, on the drug. So easy route of administration is one thing. It's very safe. The most frequent side effects are injection site reactions, grade 1, grade 2. So from a patient compliance point of view, I think it can play a very nice role. And of course, at the end of the day, besides being well tolerated, it's about the efficacy. And there, what we saw is, I believe, pretty encouraging. In different indications such as head and neck or non-small cell lung cancer or metastatic breast cancer, and is a pretty robust fundament, I believe.
FD has potentially broad applications. How have you gone about prioritizing the indications you're going to pursue?
Yeah, that's also a good question. So indeed, when you are looking at the different indications as a biotech company, you have to make some choices. You can't do everything. And even within one indication, you have different options. So you need to make some choices, and we try to look at this in a very objective way. So you look, of course, at the potential benefit which could be provided to the patients. You look at the feasibility, so how many patients you could recruit. You look at the competitive landscape. You look at timing, you look at resources, cost. Of course, you look also at the commercial perspective, and then you come to conclusions and possibly prioritize one approach or one indication or one subset of patients versus another.
In the case of FD, where has that led you?
Yeah, that's a decision we made relatively recently. Indeed, FT is a very versatile, practically pipeline in a product. So we address obviously the patient's immune system, and this could work in a variety of different indications. We have different clinical trials or performed different clinical trials in the past. We have seen data. So we needed to make some choices, and we believe that the first priority is in first-line non-small cell lung cancer, especially on the basis of data we relatively recently presented at the ESCO in an oral presentation. Also, we will continue to prepare for late-stage clinical trials in metastatic breast cancer. But if you need to spend $1, and this depends, as described, on many factors. Another factor is, of course, also the key opinion leader environment, other strategic stakeholders in the game. But then you try to maximize, and this would be, even though it's a very, very big indication, of course, also with challenges, but in first-line non-small cell lung cancer.
What's known about EFTI from the studies you've done to date?
Yeah, so we have different data points. First of all, what I said earlier on, we activate the patient's immune system, activate the APCs, and then via the physiological way, more CD4, CD8, more NK cells, Th1 response. This is actually something you can measure in patients. In May this year at the ESMO breast cancer conference, we presented some biomarker data out of the so-called APEC clinical trial in metastatic breast cancer. It's a randomized, double-blind, controlled clinical trial looking at paclitaxel plus eftilagimod versus paclitaxel plus placebo. And there we actually measured in patients a significant increase, for instance, in terms of CD8 T cells, in terms of activated monocytes. We saw an increase in CD4 T cells. We saw an increase in terms of the chemokine CXCL10. So we were also able to link the improved immune status to improved overall survival. So there's a statistically significant correlation. So first of all, we see that the hypothetical concept boosting patients' immune system is something you can measure in patients. So that's, of course, very important, especially in a controlled clinical trial. Second point is that we have a good safety profile, so that we are not adding, for instance, what you see with many immune checkpoint inhibitors, that we are not adding to the onset of autoimmune diseases, treatment-induced autoimmune diseases. So we do not see that. I mentioned already the injection site reactions, but I think that's very manageable. And then we see efficacy. For instance, in the combination with anti-PD-1, pembrolizumab or Keytruda, whatever you prefer, it's a term, we saw in first-line non-small cell lung cancer as well as in second-line head and neck cancer that we are able to, for instance, double the overall response rate, and that we can also achieve responses in patients which would typically not or suboptimally respond to an anti-PD-1 therapy alone. So this was pretty nice and has been leading, for instance, to an oral presentation at ESCO, obviously the worldwide biggest and most important clinical oncology conference. We are very excited that we had the chance to do that, but of course we are moving forward, and there will be hopefully other exciting data points in the future. We saw also a good signal in melanoma, in second-line head and neck cancer, in metastatic breast cancer. And that's fairly important because marketing statements are one thing. And of course, you would try, if you can, based on facts, to talk about your concept, but the data needs to be there, the fundament needs to be robust, and you need to have a chance to position yourself in the concert of existing standard of care and also in the light of other late-stage developments.
Now, what's the development path forward?
Yeah, so we have different potential clinical trials. First of all, we are conducting a randomized trial in first-line head and neck cancer together with US Merck, which is good. We have been teaming up with the big pharmaceutical industry repeatedly. So they provide Keytruda. We conduct the trial. It's a trial looking at first-line head and neck cancer, randomized. So Keytruda plus eftilagimod versus Keytruda within the label of Keytruda, plus satellite cohort where practically you can't measure PD-L1, so-called CPS score, where we give also the combination, but it can't be controlled because no PI would give an anti-PD-1 therapy there. So this is ongoing. It's currently recruiting. We recruited now around about 30% of the patients, and we hope that we have the recruitment finished mid of next year, and then we would see also, of course, first data. This trial is based on fast track designation by the FDA. So that's quite important. We are also continue to treat patients in our TACTI-002 clinical trial in first-line non-small cell lung cancer, second-line non-small cell lung cancer refractory to PDX treatment, and second-line head and neck cancer. It's fully recruited, but of course there are still some patients under treatment. So we perform that trial. We relatively recently announced a new trial in soft tissue sarcoma, which is an investigator-initiated trial from the Marie Curie Center in Warsaw in Poland. It's fully funded by the Polish government, and it's looking at the neoadjuvant setting, including radiopharmaceuticals. So it's a new setting, and it's of course quite exciting. And you will possibly see us also moving forward, initiating new clinical trials or enter into new collaborations in order to horizontally roll out the eftilagimod case, meaning to detect additional signals in different other indications.
Immutep has a number of collaborations, notably with GSK and with Novartis around different candidates. What's the general approach Immutep has taken on partnering? How do these different collaborations fit into your overall strategy?
For us as a biotech company, the big pharmaceutical industry is the natural customer. This is a very typical business model for most biotech companies. We have currently no commercial capabilities. If that would change over time remains to be seen. But for biotech, very, very often big pharma is a customer. And you should know your customer, irrespective of the industry you are working in. You need to be exposed to, for instance, criticism if they don't like a concept or if they don't like a trial or whatever. So we were always a fan of discussing with them relatively early on to team up, to pass due diligences. Of course, with all confidentiality which is attached to that, and of course with protecting your own position. But it's important that you incorporate your potential customer and the guys who are active on the market every day into your plans and into your development strategy, because we develop our drugs towards the market. We would like to see that our approaches can make a difference for patients. And in order to achieve that, it's good if you team up. And we have collaborations of different natures. So we have exclusive worldwide licensing agreements with GlaxoSmithKline, with Novartis. We have a territorial licensing agreement with EOC Pharma for eftilagimod in China. We have clinical trial collaborations with US Merck, with German Merck and Pfizer, where we collaborate on a specific clinical trial. So I believe it's very important. And it's, of course, not a secret. Also in every industry, if you have potential number of competitors, if what you are doing is delivering good results and it's getting more and more interest, then you can potentially also achieve a better price if you have a number of bystanders and interested parties.
Well, walk us through each of those collaborations. Let's start with Novartis.
Novartis is our exclusive worldwide licensee for irinotecan. That's an anti-LAG-3 program. So it's, if you like, the mainstream of the LAG-3 development. So anti-LAG-3, typically in combination with anti-PD-1. Novartis is conducting 5 clinical trials. And they published some results. And we hope, of course, that they publish some more results in the future. It's a milestone and royalty-bearing partnership. With GSK, to go to the next one, it's also an exclusive worldwide licensing arrangement. GSK in-licensed GSK 781. It's a depleting antibody positioned in autoimmune diseases. GSK 781 is a cytotoxic antibody designed to destroy the chronically LAG-3 activated T cells. It has been moved through preclinical development by GSK and has been successfully tested in 2 phase 1 clinical trials, one in healthy volunteers and psoriasis patients, where the biology has been tested and has been also measured and confirmed. One trial in healthy volunteers and Japanese and Caucasian patients. They then jumped to a phase 2 in ulcerative colitis. That trial had been stopped and discontinued, and we are waiting for next step. The licensing agreement has not been terminated. Also, we are waiting for next steps and potential new areas of development for IMP 761 or GSK 781.
It's been a tough market environment for public biotechs. Immutep is trading at less than half its 52-week high. How far will existing cash take you, and what's the plan for raising additional capital?
Yeah, first of all, you are absolutely right. The biotech markets, equity capital markets are, of course, difficult. We saw a drop, for instance, in the XBI, the relevant ETF, from, I don't know, 140, 150 to now 78 or 80. So a lot of biotechs, especially in the small cap, have been losing, unfortunately, a great deal of value, but you have to live with those times, and you need to execute your business and make a difference. It's not the first crisis we see in the markets. We have a seasoned management team. We have always been very strict in terms of our budget. There is no money to waste. It's money our shareholders and investors have been entrusting us with. So it's our job to deploy that hopefully wisely and to get the best value out of the dollar invested. And this starts with every employee, all the expenses that we negotiate quite hard. Also an environment where you see inflation kicking in, where you see maybe more difficulties in terms of supply chains, that you have your business under control. So our cash life is currently till early 2024. We have been in the past, and we continue to do so, budgeting conservatively. And we are, of course, fully aware of the market conditions. We are not stressed about raising cash in the short term. I believe we have a number of different strategic options. We have different possibilities. We have been getting a great deal of support from our institutional investors, substantial holders, for instance, Fidelity. We have a very good Australian and US and Asian and European institutional investors. We have a number of retail, so-called mom and dad investors, for which we, of course, or for whom we have, of course, very high responsibilities, because these investors can't be immunologists or not even experts in oncology, which means that we have to transparently communicate. But I believe we have a pretty realistic view in terms of the market. I believe that LAG-3 in general is in favor. And yes, our share price suffered as well. That's true. But we have a lot of potential, and I believe we have an exciting fourth quarter of this year, which is actually running. So we plan to present at SITC in Boston, 8th to 12th of November. So there is a lot to expect from us. And I think if the share price is lower, and what we believe maybe not so much reflective of your value, in turn it believes that it might be an opportunity for the one or the other investor. But main thing is that we continue to deliver, that our basis, the data, our manufacturing efforts, our regulatory affairs, that this is in good order and that this is delivering and showing a robust fundament for our products and the company.
So forgive me, how much runway do you have, and is there a plan to raise additional capital here?
Currently, no plans. We have cash runway till early 2024, and no actual plans to raise cash.
Marc Voigt, CEO of Immutep. Marc, thanks so much for your time today.
Thank you. It's been a pleasure.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.